Imagine you are a food detective, equipped with an array of tests designed to uncover the secrets hidden within your meal. By using simple yet clever biochemical methods, you can identify the various types of molecules that make up the food you eat. Let's embark on this investigative journey and learn how to detect carbohydrates, proteins, lipids, and more. Benedict's test for reducing sugars. Reducing sugars, such as glucose and fructose, can be identified using Benedict's solution, a blue copper C sulfate reagent. The procedure for this test is 1. Mix a small amount of the food sample with Benedict's solution in a test tube. 2. Heat the mixture in a boiling water bath for 2 to 5 minutes. Observation. A color change from blue to green, yellow, orange, or red indicates the presence of reducing sugars. The intensity of the color change corresponds to the amount of reducing sugar present. Iodine. Tests for starch, a polysaccharide, can be detected using iodine solution, which is brownish yellow. The procedure for this test is 1. Add a few drops of iodine solution to the food sample. Observation. A blue-black color indicates the presence of starch. If no color change occurs, starch is absent. Testing for proteins, burette. Test proteins can be identified using the Biuret reagent, which contains copper sulfate and sodium hydroxide. The procedure for this test is 1. Mix the food sample with an equal volume of Biuret reagent in a test tube. 2. Gently shake the mixture. Observation. A color change to violet or purple indicates the presence of proteins. The intensity of the color change reflects the protein concentration. Testing for lipids. Emulsion. Test. Lipids can be detected using ethanol and water. The procedure for this test is 1. Mix the food sample with ethanol in a test tube and shake well to dissolve the lipids. 2. Add water to the mixture and shake gently. Observation. A milky white emulsion indicates the presence of lipids. The formation of a clear layer means lipids are absent. Testing for vitamin C, DCPIP, test vitamin C or ascorbic acid can be identified using DCPIP, dichlorophenol indophenol, a blue dye. The procedure for this test is 1. Add a small amount of DCPIP solution to the food sample. Observation. The blue color of DCPIP will disappear, turn colorless, in the presence of vitamin C. The more vitamin C, the faster the decolorization. Testing for complex carbohydrates. Barfold's test for monosaccharides. Monosaccharides can be identified using Barfold's reagent, which contains copper 2 acetate. The procedure for this test is 1. Mix the food sample with Barfold's reagent in a test tube. 2. Heat the mixture in a boiling water bath for 1 to 2 minutes. Observation. A red precipitate indicates the presence of monosaccharides. Combining these tests, you can paint a comprehensive picture of the molecular composition of food samples. Here are some clever approaches. 1. Sequential testing. Conduct tests in a specific sequence to identify multiple components. For example, start with the iodine test for starch, followed by the Benedict's test for reducing sugars, and then the Biuret test for proteins. 2. Control samples. Use known control samples alongside unknown samples to compare results and ensure accuracy. For instance, use a glucose solution to validate the Benedict's test. 3. Quantitative analysis. Estimate the concentration of molecules by comparing the color intensity or the amount of precipitate formed. This can provide semi-quantitative information about the food's nutritional content. 4. Microscopy. Complement chemical tests with microscopic examination to observe structural details such as the presence of starch granules or fat droplets. 5. Paper chromatography. Separate and identify small molecules like amino acids or simple sugars using paper chromatography, where different molecules migrate at different rates on a paper medium. These food tests have practical applications in various fields. Nutrition and health. Determine the nutritional content of food, ensuring balanced diets and identifying nutrient deficiencies. Food industry. Monitor food quality, authenticity, and safety by verifying ingredient composition and detecting contaminants. Education. Provide hands-on learning experiences in biology and chemistry classes, helping students understand biochemical principles. Forensics. Analyze food samples in criminal investigations, such as cases of food adulteration or contamination. By employing these clever biochemical tests, you can unlock the hidden molecular composition of food, revealing the presence of carbohydrates, proteins, lipids, vitamins, and more. This detective work not only enhances our understanding of nutrition, but also ensures food quality and safety, making it an indispensable tool in both science and industry.